Chris, can you hear me?
Oh, sorry. You want me to start?
No, I'm going to
Oh,
introduce you. I just wanted to make sure that everything is working. So welcome everyone to this seminar as part of the Menten Institute. The speaker today is Chris Soteros from the University of Saskatchewan, and she's going to talk about mathematical modeling of DNA topology. Chris, please take it away.
thank you very much, Margarita. And thank you to Mark Lewis and Stephanie Portet for inviting me to give this talk. I should start by saying I acknowledge that I live and work on Treaty 6 territory in the homeland of the Métis, and I pay my respects to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. And so Mark commented about my accuracy of the coordinates for Saskatoon. So that I started to put that because back during the pandemic, I gave a talk remotely to India, for example, and I thought people didn't actually know where Saskatoon was. I don't know the accuracy. I think I just copied it from the internet, so I didn't think about the number of significant digits. Um, what I'm going to talk about, so I guess, first of all, I have lots of collaborators to thank for what I'm going to talk about today, and supporting agencies and CERC PIMS and Digital Research Alliance of Canada. And mainly I'm going to, because first of all, this is a math biology seminar, but I'm not strictly speaking typical math biologists. And I think neither is DNA topology, a well-known area of math biology. So I thought I would start by giving a bit of my background, and also a little bit of the history to the field of DNA topology, at least from what I know of it. So I did my PhD back in the 80s, a long time ago, and I was in an interdisciplinary applied math program where I could choose a supervisor from any department. So my supervisor was Carol Hall, a physicist in the chemical engineering department, and she introduced me to statistical mechanics modeling, and in particular, back then, we had just got through the 70s and the energy crisis, and she was looking at modeling ways to store hydrogen in metals. And so I looked at models of phase transitions for hydrogen stored in metals, such as niobium. And I learned about how to use combinatorial analysis, because this is a lattice model, as well as Monte Carlo computer simulation. And then I did a postdoc. I came back to Toronto, to Canada. I'm originally from Windsor, Ontario. I did my bachelor's in mathematics. And I did a postdoc with Stu Whittington, who's a chemist, a physical chemist at the University of Toronto. And but very mathematical. And he introduced me to models of polymers, and in particular, lattice models of polymers. And really, I've been studying lattice models of polymers and models of polymers more generally since then, including and other applications of lattice models of statistical mechanics. And while I was a postdoc, Stu introduced me to DeWitt Sumners, who is a topologist at Florida State University. And so he is along with Nick Carzarelli, a biochemist from Berkeley, they started the program in mathematics and molecular biology back in the 90s with funding from Burroughs Wellcome. And they were responsible for really training a generation of DNA topologists, so students and postdocs who learned mathematics at the same time as actually doing lab work. wet lab work. And in particular, I want to highlight two of DeWitt's former students, Dr. Mariel Vasquez and Dr. Javier Arzuaga. They're both at UC Davis now. They both have joint appointments, Mariel in math and microbiology and molecular genetics, and Javier in mathematics and molecular and cellular biology. And they both continue to do both math modeling, math research, as well as actually experiments and working closely with experiments. So I work closely with Marielle, and so I'm not working directly with any experimentalists, but she does. And so working closely with her keeps me connected to the experiments. And also I've learned a lot from both of them about the general field of DNA topology. And I might as well say it up front in case I forget or run out of time to say it at the end. So there's some upcoming opportunities to learn more about the field. So in particular, Marielle will be giving a PIMS network-wide colloquium on March 20th. And then at the Statistical Society of Canada annual meeting in Saskatoon in May, I'll be organizing a small session on probabilistic models related to biopolymer modeling. Actually, Margarita will be giving a talk there. And then in June, Stephanie is organizing, amongst others, the summer school on quantitative molecular and cellular biology. And I believe there will be, one of the courses will be related to the topics I'm going to talk about today. And then in November, along with Javier and two others, I'm co-organizing a BERS workshop on polymer modeling and DNA topology. And if you're interested in attending that, please let me know. Okay, so for the most part, I'm going to be talking about the polymer models of, I'm going to be talking about polymer models of DNA. So for me, and there'd be statistical mechanics-based models. So statistical mechanics model, you put in some input of the microscopic interactions, but you're interested in the macroscopic properties. And typically, the kind of models that I'm going to be talking about, well, DNA, as we all know, has a double helix structure. Well, I'll be looking at more simplified models where we just represent the backbone of the DNA, and then even further, we might put it on a lattice. However, this is an example of a lattice walk, and if you put in enough edges, the fact that it's on the lattice is maybe not so relevant anymore. And in fact, there are known quantities that are believed to be exact for lattice models of polymers as for real polymer models. All right, so, but what is the size of DNA, or what's DNA look like in terms of as a polymer? Well, actually, for example, in the human cell, DNA in each cell, if you were to stretch it out, would be as long as 6 feet. And typically, DNA in any organism has to be highly condensed or packed into, it's very large relative to the size that it's packed into. However, and so then you might think, okay, well, is it really a polymer with the same kind of freedom that I showed on the previous page? But in particular, even though, say, in the human cell, it's packed into chromosomes, the actual DNA has to be opened out in order for cellular processes to take place, in order for transcription to occur and other cellular processes. All right, so what is DNA topology? So DNA topology is the study of topological properties of DNA in vitro or in vivo. But topology actually includes not just knots and links, but also geometry such as supercoiling and twist and writhe. So Well, first of all, you might ask, well, do knots actually occur in DNA? So this is an electron micrograph back from 1983 from DNA from E. coli. And it's been coated, the DNA has been coated with a protein. And so you can't really see the double helix. But if you were to look at this closely, you could recognize that some of them are overcrossings and others are undercrossings. So this is a trefoil knot. And I guess I'm assuming everybody knows what knots are, but mathematically, a knot is really a circle, which if you were allowed to, if you could put it in the plane without any crossings, then it would be considered the unknot. But if you can't put it in the plane without there being any crossings, and if the only way to get rid of crossings is by cutting it, then we say it's knotted. All right, but also the amount of, so in the DNA is a double helix, so there's already some twisting of the two strands of DNA around each other, but then the DNA itself can be twisted upon itself. So these are pictures of twisted DNA from AFM images from 2021 from the Pine Lab. And the amount of this supercoiling, so the coiling beyond the sort of twisting at the double helix level, is important to cellular processes as well. And so the study of DNA topology is the study of how these knots and links or supercoiling affect the properties of DNA in the cell. And in particular, it's known that knotting or linking of DNA in the cell can be an obstruction to cellular processes such as replication. So if you have, if you create, you have an original DNA, you create a daughter DNA, but if the two are linked, then when you go to separate them, that will be difficult. But the cell has enzymes which can resolve these problems. They can get rid of links, and also they keep the supercoiling level of the DNA in the cell at an appropriate level. Okay. So what are some typical DNA topology experiments? So some of the ones often referred to or go back to the early 1990s. So Sean, Wang, and Ribenkov looked at viral DNA, so phage DNA of different lengths, 5.6 kilobase pairs and 8.6 kilobase pairs. And originally it was linear DNA, and then they randomly cyclized it. And they looked at the probability of knotting in the resulting DNA, cyclized DNA. They determined whether there was knots or not by using gel electrophoresis. So different knots, depending on the number of crossings, like the trefoil or figure 8, go through the gel at different rates. And they found that they also varied the salt concentration. So DNA is electrically charged, it's negatively charged. And so if you put it in varying levels of salt concentration, low levels of salt, the DNA will be more free to move around. But as you increase the level of salt concentration, the DNA becomes more repelled from the salt ions, and it starts to become more compact, and that promotes knotting. And so you see that the knot probability does go up with the salt concentration. And more recently, in 2016, Plays et al. Actually looked at the probability of knotting versus DNA size, where instead of using gel electrophoresis, to detect knots. They put the DNA, it was actually linear DNA, in a nanopore, a solid state nanopore, where they had a charge difference between one side and the other, which drove the DNA through the small pore. And then depending on how many strands of the DNA, so if it's knotted, there'll be more strands going through the pore. And so the amount of damping of the charge, or the electrical charge, or electrical signal as you go through the pore would indicate how many crossings were going through, and they could use it to detect the knot type. And they also looked at probability of knotting as a function of different DNA lengths. So they went up to 166 kilobase pairs, and they had to have a fairly high salt concentration in order to promote their knotting. All right, so that's one kind of question of interest. What's the chance of DNA being knotted? How does it depend on salt concentration, and how does it depend on the length of the DNA? Another, as I mentioned, enzymes in the cell can control the amount of supercoiling and also the amount of knotting and linking. One particular type of enzyme is called topo2 or topoisomerase 2, where it cuts double-stranded DNA and then passes one double-stranded piece of DNA through another. And so what it's doing is, at the place where it's cutting, it might have started with the two double-stranded pieces making a crossing in one way, but after the cut and the pass-through, you've changed the crossing. And as a result, you might have started with a trefoil and ended up with an unknot. So some early experiments from 1997 due to Ribonkov et al. Actually showed that these topoisomerase enzymes, even though they're not site-specific, they don't target specific base pairs in the DNA, they're able to reduce the amount of knotting and linking of DNA molecules to, they said, 80 times lower than what you might expect from random cyclization, like the experiments that I showed you previously. So one of the areas of interest in DNA topology modeling is, what are the factors that allow the enzymes to determine where exactly to do these strand passages so they can unknot the DNA so efficiently? Because a knot is sort of trapped in the circle. It's not a local property. It's in general a global property. How can they know exactly where to attack? And then also, for example, in other kinds of enzymes, site-specific recombinases, they act, for example, during replication, and they do a different kind of operation on the DNA. They change when you have two strands, two DNA pieces together like this. They do a rearrangement and change them to be like that. So they go from this to this. It's called a reconnection operation or a strand exchange. And they can start, you could start with an unknot and then get a link out. But they are actually site specific, and they tend to act in a way to simplify the topology. Another kind of DNA topology experiment that has inspired a lot of study is looking at DNA packed in a viral capsid. So the way that it's packed in there, so that it's packed quite tightly, seems to influence. And then if you release the DNA, you allow the sticky ends to form the DNA into a cycle, then knots get trapped. And if you look at the knot distribution that you see, first of all, it's highly complex. But also you see that chiral knots, so knots that are different from their mirror image, for example, trefoil is different from its mirror image, are more likely to occur than what you would expect for just random cyclized DNA experiments. And so the knot distribution can be used to try to determine how exactly the DNA might be packed in there. And then more recently, Margarita is involved in this. People have been interested in looking at R-loop formation. So an R-loop is formed during transcription. So in transcription, you have RNA being, you have the DNA, double-stranded DNA opened, and you have RNA polymerase binding to the DNA, and it produces a copy strand of RNA from the templates, one of the template strands of the DNA. But so normally that RNA would just go away, but sometimes that RNA stays bound to the template strand of the DNA, and a single strand of DNA is left unmatched. And so you get this sort of bubble formed. And so people are interested, well, what's the likelihood of these things being formed? And it seems it does depend on the DNA sequence, but it also depends on the supercoil level of the DNA. And then also there's AFM pictures of what these R loops look like. And so there's questions about, okay, is the single strand DNA wrapping around the hybrid part, or what other things might be going on? Okay, so that's sort of a quick example, and certainly not exhaustive, of the kinds of experiments done on DNA that have inspired DNA topology questions. And of course, there's also a range of DNA topology models. So you can look at the atomistic level, and you can model all the atoms in the double helix. And that has been used quite effectively by Lynn Ziedrich and other co-workers of hers to show that, for example, if you underwind the DNA, as in this picture, so normally DNA is negatively supercoiled in the cell, or tends to be negatively supercoiled in the cell. But if you underwind it a lot, then it opens up the DNA, and you have base pair flipping. And so that actually could be a good thing. You might want that, because if you want a protein to come in and do something, you do need the DNA to open up. And so DNA topology is, in terms of supercoiling level at least, is important for cellular processes to proceed. But of course, these kinds of models have their limits, in that you can only do small DNA with including all that information. So at the macroscopic level, more like what I was introducing at the beginning, are lattice models and off-lattice models, and sort of what's become sort of standard off-lattice model for modeling DNA is the worm-like chain model, where there you have rigid rods representing with a certain diameter, and you allow certain bending angles, and you can put in interaction energy, and that has been quite useful for modeling DNA. And then there's a even higher level, sort of abstract level, where you represent what's going on by diagrams. So for example, this is a picture, sort of a knot theory picture at a diagram level of the action of topoisomerase, or topo2, or this kind of picture for modeling recombinase. And so I guess where lattice models are useful is because they're discrete and they're on the lattice. And as I will show you, that some problems become more tractable. You can do combinatorial analysis. They form a bridge between these abstract mathematical diagram level models and perhaps more realistic models like worm-like chain, but also, as I will show, that the worm-like chain models also have a lot of assumptions, and it is possible to get a lattice model that gives results comparable to worm-like chain. And there's, of course, this missing gap in between the connecting the atomistic level and the macroscopic level. I'm not going to talk about that, but that is a big area of interest, and there are people working in that in-between region. Okay, so that's sort of a quick overview of what DNA topology is. So I want to talk about a few things that we've been working on or we've made progress on recently, but I do want to give you sort of a bit of a background of why lattice models are useful and how they have been used in the past. So in particular, some DNA topology related questions that have been studied with lattice polygon models, but also polygon models in general, is first of all, is knotting linking in polymers inevitable? And this, I'll show you in a minute, was first proved using a lattice polygon model by Sumners and Whittington. And then another question is, well, what is the simplest ingredient model for DNA knotting probabilities? And I will say, so worm-like chain models and lattice models have been used since 1993. And I will say a bit more about some work with Matthew Schmerler on that. And then what model features control knot link distribution observed? And there's been lots of different studies of that. I will focus on some of our recent results related to when DNA is, or when polymers are confined, how does it affect the knot distribution? I'll be looking at a specific nanochannel type confinement, since the recent nanochannel experiments have got people interested in that kind of confinement. Okay, so basically, what's a lattice model? We assume the polymer conformations are represented by self-avoiding walk or polygon on, I'll stick to the simple cubic lattice, so the Z3 lattice, but there are other lattices, three-dimensional lattices people can consider. We usually assume that you have dilute solution, so that really you're looking at one molecule, one large molecule isolated. And we assume that we have equilibrium, so two equal energy, size, and conformations are equally likely. And we also assume two conformations are the same if one is a translate of the other. So, for example, if we just assume that each polygon, we look at the simplest sort of model, say each polygon of the same length on the simple cubic lattice is equally likely, you could ask, what's the probability that it's knotted? So there, that becomes a counting problem. So for a fixed number of edges, you'd want to know the number of polygons that were knotted divided by the total number of polygons. So that would be the probability of being knotted. Or typically what you do is you count the number that are unknotted and you subtract that from 1. So but on the simple cubic lattice, for example, you can do exact enumeration, but the best counts seem to be up to length 36. And you need at least 24 edges to get a knot. Oh, that's for walks, right? For polygons, it's 32. You need at least 24 edges to get a knot. And you don't really have very many knots yet at 32 edges for polygons. So this exact enumeration approach doesn't really get you very far. All right, so, but there are other ways that you can prove things. So going back to are knots inevitable? So back in the 60s, Frischmann, Wasserman, and Delbruck conjectured that if you had a sufficiently long polymer, it would be knotted with high probability. And this was proved first using lattice polygon model by Soteros and Whittington in 1988 and independently by Pippinger. And there they used what's called a pattern theorem argument, and they focused on the case that each polygon was equally likely. So let Pn be the number of n-edge self-voiding polygons, Pn01 the number that are unknotted, Pnk the number that are of a specific knot type K. Then, as I said, the probability of knotting would be 1 minus Pn01 over Pn, and they proved that it goes to 1 exponentially with the length of the polygon. And they proved it by using a pattern theorem for polygons, and they showed that all but exponentially few sufficiently large polygons contain this sort of tight trefoil pattern, which if it occurs in the polygon, it ensures that the trefoil is in the knot decomposition. And then back in the early 90s, myself with Soteros and Whittington, we proved that the probability of any given knot type has to go to 0. But there was many open questions. So by using Monte Carlo studies, it was conjectured that the number of polygons and edges with knot type K has this following scaling form. Grow like unknots, but with n to the fk factor, where fk is the number of prime knots and k is knot decomposition. And the basic sort of idea behind the conjecture is that it's believed that knots, when they occur, are fairly localized in the polymer. So if you have a large polymer with lots of freedom and it's a trefoil, that the trefoil part is relatively small. And so the number of trefoil polygons could be obtained roughly like starting with the number of unknot polygons and then choosing one of the edges to fit in a little trefoil pattern. So that's the idea behind the argument. So it's believed that knots are localized. I don't think I have time to go through that. So, but this is just illustrating that the combinatorics behind the Soteros and Whittington proof. And so similar things have been studied for systems of polygons. So for example, if you have some polygons in a box, you might ask, what's the chance that they're linked? And so you cannot show that if you have to put some constraints on the box size, but if your polymers are length n and you let the box size be the same length as the polymers, then you cannot guarantee that linking occurs, at least not, you can't show the same way that you did for knots. You can't show that it goes to 1 exponentially. Probability may still be 1, but it won't be 1 exponentially. But there's some recent numerical results that show that you do expect links when they occur to be, just like knots, to be somewhat localized. So if you if the lengths of the 2 polygons are not fixed, you allow them to be variable, then you'll have a small link, for example, being localized just the way you would having a trefoil localized. All right, so this is a combination of things that have been proved for self-voiding polygons on the simple cubic lattice and the knot distribution, and also things that are believed to be true based on numerical results. So let's go back to the experimental knotting probabilities that from the Sean Wang and Ribenkov experiments, where they randomly cyclized DNA of certain lengths and looked at how the knot probability changed with the salt concentration. So these are the graphs from two papers. This is from Sean Wang for 8600 base pair DNA, and this is from Ribbon-Cobb et al. For, this is the percentage of trefoils. This is the knotting probability for 11.6 kilobase pair DNA. So it does increase with salt concentration. So back in 94, Tessier et al. Did propose a lattice model for modeling this knot probability, where they, instead of taking each polygon to be equally likely, they weighted polygons by a combination of the number of what they call contacts, so edges between two non-bonded pairs in the polygon, and an electrostatic term, which has a long range, which is a long-range interaction, an interaction between vertices in the polygon that are different distances apart. And they could get different conformations depending on the salt concentration. So at high salt concentration, you do see that the polygons are much more compact, and at lower salt concentrations, a little much more open. And it has the same number of adjustable parameters as this discrete worm-like chain. So people didn't look at this lattice model because the worm-like chain model sort of took off as the most popular model. People didn't really look at this very much. And in the Tessier et al. Paper, they found parameters for their model that did give a knot probability distribution similar to the Sean Wang experiment, but they didn't do a thorough sort of fitting of the model. And so Matthew Schmerler, my PhD student, former PhD student, now postdoc, for his PhD work, he looked at developing a way to fit the parameters in the model to the Sean Wang DNA knotting probability experiment to try to get an improved model. And he first started by looking at the original model that Tessier et al. Looked at, which had the nearest neighbor, this contact interaction, as the local interaction, as well as the long-range electrostatic term. And then he also looked at another model, where it disfavored right angles. So this is called bending rigidity, and weighted that for local interaction instead of the contact. So this is because DNA is a relatively stiff molecule. It's what they call semi-flexible because of the double strand, and the double helix makes it somewhat stiff. And the contact model didn't seem to be getting that kind of stiffness. And so Matthew then used what's called, to get the knotting probabilities for different parameters, choices for his model, he used Markov chain Monte Carlo and a 2-point pivot algorithm, where you start out with a particular polygon and then you make a change to it to go to a new conformation, and you weight the change according to the Gibbs potential. And then he used stochastic approximation methods, essentially a stochastic optimization method, to fit the model parameters to the DNA knot probability data. And there the idea is, so it's called simultaneous perturbation stochastic approximation. So you start with some initial choice for your model parameters, and then you choose several directions for your parameters. You calculate using the Monte Carlo, you estimate the knot probabilities for those different parameters, and then based on the outcome, you choose a new set of parameters. So it is known that this will converge, and so he did get best parameters for the bending Yukawa model. That's the one that had the rigidity term, length, polygons of length 600, Those are the red ones. And for length 800, the contact Yukawa model, those are the square ones. We got comparable fits to the DNA knotting probabilities from the Shaw and Wang experiment. But one notable difference is the conformations that these polygons can take on. So with the contact Yukawa, I showed you some examples before at this is, I think, physiological salt concentrations. You have sort of this open shape, but you've got sort of all these little right angles and everything on a small scale, where with the bending Yukawa model, it's open, but it shows this very rigid, and indeed, something called the persistence length is closer to what's expected for DNA with the bending Yukawa model than with the contact Yukawa model. So, and then he used his best bending Yukawa model to compare to some worm-like chain simulations for different DNA lengths at physiological salt conditions, and basically predicted almost exactly the same. It's hard to tell the difference between the 2 points on this graph, one blue and one red. So that the model so far is doing, at least in terms of knotting probability, just as well as the worm-like chain model. So summary of that work was we used coarse grain model of relaxed randomly cyclized DNA and salt solution, and the bending DNA Yukawa model captures DNA persistence length better. But we would like to get better resolution by increasing the polygon length and also use the model to compare to other experiments. So I know I'm running, I only have about 5 minutes left, but I do want to say a bit about the other sort of progress that we've made by looking at modeling DNA in nanochannels and nanopores. I told you there about the experiments, but they were using nanopores to identify DNA, but people are also looking at DNA conformations in nanochannels as well. So here we've used, we're looking here at polygons in confinement, but now in what we call tubular confinement, still on the simple cubic lattice. And the type of question we can ask is, well, are knots still inevitable? And what about for the people expect for the unconfined case that the knots are localized? Are they localized in this tubular confinement? And so, in fact, by looking at the tube model, we can actually prove things that only are conjectures outside the tube model. So in the tube, we consider, for example, a polygon where we've restricted, say, the height to be m and the y direction to be restricted to L, and we allow the polygons to grow in the x direction. And this has the advantage that we can basically think of a polygon as a word made up of pieces, the pieces that can fit on these sections. And because of that, we can count the number of polygons or even get a generating function for polygons weighted by certain properties using transfer matrices. So where this is just a 2D example, where the transfer matrix incorporates, say, the number of ways to go from this sort of letter, which is on a section, to this letter, and where we put in some factor that to keep track of things we want to count. And so you can use the transfer matrix to get partition function or a generating function, depending on whether you're a statistical mechanics person or a combinatorics person. And you can get asymptotic properties. So you can get, for a statistical mechanics person, results about the free energy. For a combinatorics person, the exponential or asymptotic properties of the counts. And we can also prove pattern theorems. So you can prove that the FWD conjecture, that all but exponentially few polygons in a tube are knotted, by showing that they contain some knot pattern. But you can prove even more. You can prove that the expected number of occurrences of a pattern grows linearly with either the tube size or with the number of edges in the polygon. And also it gives us a nice Markov chain way to generate polygons in the tube. And so the transfer matrix can't tell you whether your polygon is knotted or not, because that's a global property, not a local property. So we still need to generate polygons, but the transfer matrix gives us a way to, okay, we start with the beginning of the polygon, and then we can choose randomly from amongst the letters that could be connected to it, and so on, and we can generate polygons, and then we can look at the knot type. And so we've studied it numerically, and the numerical results do suggest that knots are localized in the polygons. And most recently, in the 2x1 tube, we are actually able to prove that that is true. So in that 2x1 tube, it's the smallest tube that emits knots. And so we were able actually to prove a pattern theorem for unknot polygons, which this is something that's not been proved for any other model. So because we have a pattern theorem for unknot polygons, we can turn an unknot polygon into any knotted polygon we'd like. And then the other thing that was able to prove with my knot theory collaborators, Koya Shimakawa and Kai Ishihara, that if you start with a knotted polygon in the 2x1 tube, you can find a place to unknot it without changing it very much. So I think more or less stop there. I'll just say for the nanochannel models, we've able to prove results for 2x1 tube. And what's next, regarding we maybe would like to use the bending Yukawa model to add interactions and see how that affects our results. And also with Margarita and Marielle and Natasha Yanovsky and other collaborators, we are exploring applications to modeling the DNA RNA hybrid, such as the R loops that I mentioned earlier. So finally, thank you for your attention, and I'll remind you of some upcoming opportunities to learn more.
Thank you so much, Chris, for this nice talk. If you have any question, please feel free to write in the chat, or you can unmute and ask directly to Chris.
I know. Oh, somebody's got their hand up.
Yeah.
Yeah. Yeah, thank you for that awesome talk. That was really interesting. Can you, I'm sure you probably like touched on it, but can you explain again why it is so much easier to get all these estimates and theorems in like the tubular form compared to just like a confined box?
Yeah, it's okay. It's all right. It's because we're confining only two directions. So we're confining this and this. We are still allowing them to grow in one direction. All right. So it's not completely confined, because that's actually a really hard problem if you confine all directions. So why this is why this is easier than both the whole lattice, the unconfined case, and the fully confined case is this idea that I can basically think of, it's a linear, it's just growing linearly. So I can think of it as a word where, you know, I look at the conformations that can happen at a slice, and I can list them all, given a certain L and M, right? These will be fixed, okay? In principle, I could figure out what all the things that could happen in a polygon on that slice are. And then I can figure out which ones can go, which ones can follow which other ones, okay? So basically, I've got this alphabet, and I only need to know what the previous one is to know whether the next one connects, all right? So I basically got I can store in this transfer matrix basically all the information about the number of, or the ways to generate polygons. Okay, so we've turned it into, even though it's a 3D problem, it's essentially a linear problem. Now, in practice, of course, the number of these possible slices grows if you grow the L and M, right? So we are able to do exact calculations up to, like, we can do 2 by 1, 3 by 1, 3 by 2, 2 by 2, 4 by 1, 5 by 1. You could probably go a bit further, but after that, it starts to be impractical to actually construct the transfer matrix. On the other hand, there are things that you can prove theoretically because you have this sort of Markov. You only really need to know the previous one to know what the possibilities are for the next one. That because you have that, there's things you can prove. Although I should have pointed out, our proof for the pattern theorem for unknot polygons in the 2 by 1 tube, it actually relied on the fact that we had the transfer matrix for that tube. So we needed to know something about, we needed to have bounds on the exponential growth rate of unknot polygons, which we could get. Even though you don't have a transfer matrix for the unknots, because it's a global property, because we had the transfer matrix for all polygons, it gave us some bounds that we needed for our proof. I don't know if that answers your question.
No, yeah, thank you. That's very insightful.
Ah, Javier.
Yeah. Yeah.
I'm in trouble now.
No, thank you. Thank you for your talk. I have a couple of questions. So,
I might turn it around and ask you. Okay, go ahead.
So there are a couple of some experiments that show that when you put DNA in high salt, they form toroids.
Right, right.
Look at the distribution of knots in your simulations, and have you look at the shape of those knots, kind of overall shape, to see whether they...
Not yet. I think Matthew was here. I don't think we've looked at that, but I think we would need to put in some other interaction to get that. I think you and I have talked about that before. Like we might need some kind of cholesteric interaction or something, right? To force that kind of bending.
So have you thought about adding that interaction? And it might depend on the type of...
I've thought about it, but we haven't got to implementing it. So okay. I want to talk to you more about it. Yeah.
Okay. And have you thought about changing the type of ions that you are working with? Like people, there are simulations for an experimental data for like sodium and magnesium, but then there are experimental studies for spermine or kind of more heavy ions.
Yeah, so is Matthew still here? He's not here. So my understanding is in the Yukawa potential, you can actually specifically take into account the ions that you are using. So like he put in what it would be for sodium chloride, but I think that it's just changing a number there that to reflect the different kind of ion.
Okay.
So, but we haven't done it. But I know, like, I noticed the Plaza, they use lithium chloride, I think, or something. Anyway, yes. So I think we should look at it. Yeah. But I think we'd have to, yeah, so that's a question. Can we use our fitted parameters and change the ion and still get good results? Or do we have to refit? I don't know the answer to that.
Okay. And then I had one question, which I don't know if it makes any sense or not, but let me ask it anyway. So once you have, so you generate the transfer metrics, and then you can look at whether you have a knot or not, and so on. Now, can you go back to some extent saying, now I have all these transfer matrices that I know they correspond to unknots, or these transfer matrices that correspond to trefoils? Can you get any properties out of those particular subsets of matrices?
Well, no, because the resulting polygon that has the knot in it is really an entry in power of the transfer matrix. So it's just one entry in the, so it's one sequence of those letters, right? So, but there's something in your question, but I can't.
Okay, don't worry, we can talk about.
Yeah, right, okay. But there sort of isn't really a single transfer matrix associated with one knot.
Mm-hmm.
But, you know, on the other hand, what we have looked at is, you know, the knot patterns that show up in our polygons, right? So the most likely patterns would be the smallest knot patterns. And so that you can know what the density of any given knot pattern is. Once you've specified what the pattern is, you can know that from the transfer matrix, right? So, but there's, you know, lots of different knot patterns. So, okay. Oh, Stephanie.
Yeah,
I don't know who's next.
I think Clotilde.
Okay.
The question.
Yes. You can go.
Hello, I'm with the U of M watch party here. Oh, is there?
I can hear you, a bit of echo, but go ahead.
All right. So I'm just wondering regarding the knotting. So if we take just a regular unknot, you know, experience will tell us we can mangle it all a bunch. I'm really sorry for the echoing. That's bizarre. Oh, is that better? Okay. Can you
Yeah,
hear me
that's
still?
better. Yes, that's better.
Okay.
Oh,
All right. So if we take an unknot, we can wrap it around itself, do a bunch of things, and it's still technically an unknot, right? But it kind of, you know, to the naked eye would look like maybe it's a little knotted, right? But it has still has some of those like physical constraints that would affect it maybe in a similar way biologically to what these knots are actually doing. And basically my question is, can we have a situation like that where it's not actually technically a knot, but we can apply some of these techniques to it because it's a similar sort of spatial kind of constraint that is arising from that. I don't know if that makes sense.
so I'm not sure. Are you talking sort of about supercoiling, like in these pictures?
Yeah, that's definitely part of it. Yeah, I guess. Yeah,
So
I guess.
we can put in terms into the interaction energy that would force those kind of things to happen. So these are sort of local things that you're forcing to happen. Yeah, I'm not sure if you.
Yeah, no, that
Okay.
that makes sense. Yeah, I forgot about the supercoiling aspect, but yeah, that would answer it. Thank you.
Okay. Unless you were talking about linear DNA, then of course you can have anything happen.
That's definitely one thing I was thinking of.
Yeah, but then of course the knots
this
can
was
disappear
more-
on their own, right?
Yeah.
So, okay. I think now Stephanie might be next. I don't know. No? Okay.
Hi, Chris.
Hi.
Thank you for the talk. I think you mentioned that, but what is the persistent length of DNA? And I think so. At least for the simulation, you gave that in pair base pair, but in matrix in nanometer.
Oh, Matthew or Javier, do you know offhand? What is it in nanometers? I don't know offhand.
It's 150 base pairs.
Yeah, so how many base pairs per nanometer? I can we can find out. I just don't know off the top of my head.
Okay, okay. So just to compare to the polymer I'm working on, and because I know just in matrix for the persistent length, not in base pair. Thanks.
Okay.
Any other question for Chris? So if there are no other questions for Chris, we can thank her again.
Well,
for
thanks
the talk.
everybody.
And I guess the next seminar will be end of February, sort of. Stephanie, you would know better probably.
I don't know anymore. I think we have somebody. Alison, I think we might have somebody in Victoria. Don't remember anymore. I think the next one is oh mama, I don't know. Sorry, it's too late. It's 10 p.m. in Paris. I'm in Paris. Sorry. Okay. Bye-bye.
Nice to see you, Stephanie.
Bye.
Thanks for coming, Richard.